Okay, so the next speaker is David Sabatini. David doesn't really need any introduction to the MIT participants here. I think that basically David's name is kind of synonymous with mTOR because when he was a graduate student with Saul Snyder, he first identified mTOR as the target of rapamycin, and since then has really done, really unbelievably beautiful and beautiful work and been highly productive. And so look forward to your presentation.
All right, let me need the password here. You've already heard it. I know, but this is not working. Just like close a few bits. So thank you. This is on. Thank you, Joan, and also thank you, Mike, for the invitation. Also, thank you to CST for hosting us and sponsoring this. Actually, I've worked a lot with CST. In fact, I think lots of our success on the mTOR pathway is due to them. They have dutifully made antibodies that we have exploited to really every component of this pathway before we actually even showed the components mattered. So I don't have an excuse for this talk. I had no issues with PowerPoint. I did just lecture, though, to MIT undergraduates on the nervous system for about an hour and 40 minutes. And so there is a part of my brain that remained there, so you'll have to excuse me a bit. So as Joan mentioned, we do work on the mTOR pathway, and many of you know this pathway. This is a major regulator of growth, really, at all these different levels. We started with cell size regulation, but we're certainly slowly working our way in this direction, a little bit of organ size control, and increasingly thinking about body size regulation, although I'm not going to talk about that today. And the origins of this pathway, I think all of you know, starts with this molecule, rapamycin, really a very interesting molecule, both from the basic science and clinical point of view. Starts out in this little island, Easter Island, where it's a bacterial product first identified there. A number of different clinical uses, but the observation that really took rapamycin much more in sort of the public's mind is the finding that in multiple different model organisms, this molecule prolongs lifespan, and most notably, really, in middle-aged mice. And this has now been reproduced by a number of different groups. And I would say that likely this is, I think, the strongest evidence for an FDA-approved molecule having this property. In fact, I've mentioned to some of the people who I go to lots of meetings with that I've been criticized at times by actually putting this word here, immunosuppressant. I've been told that that's going to dissuade people from actually using rapamycin to prolong their lifespan, and so I should remove this. But nevertheless, it is an FDA-approved immunosuppressant, so it seems like an odd way of thinking about it. In any case, this is a pathway that's very complicated. This is an old slide on purpose, just to show you the main points. There's a main target, mTOR, which is directly bound by rapamycin. This is a kinase. And the parts of this pathway that have kept, at least my interest since I was a graduate student to now, you could argue just very unadventurous, or there was actually something interesting to study here. But the part that's really retained our interest is the fact that this pathway, unlike really the vast majority of pathways, has the capacity to sense everything. And literally, I mean everything. So any kind of growth factor, any kind of nutrient, any kind of stress, DNA damage, osmotic, somehow this system recognizes it, mTOR is regulated, and that tells us that the cell cares a lot about regulating this system. In addition, mTOR regulates really all of the major processes in the cell that either use or make energy. And so this is a key pathway in regulating cell physiology, and that's why we've been interested in it. Now, as we go much more into the in vivo side, we started thinking about this pathway in a way that I think Joan really illustrated in some of her work, is we really think about this pathway as connecting food, either manipulated by things like caloric restriction or obesity or different types of diet, to the balance between anabolism versus catabolism. That's at least conceptually how we think about it. We consider all of these things, nutrients, but then things like insulin, which fall in the growth factor category, as surrogates for the availability of food. And you'll see when I show a little bit of in vivo evidence that at least what I'll show you is consistent with that. So in fact, there are two pathways, mTORC1 and mTORC2, which many of you know. They're both surrounded by a variety of different mutations that are found commonly in cancer. And increasingly, there's actually some mutations now found in mTOR, which are turning out to be quite interesting. So there is a connection to cancer, although today I'm not going to talk about this. I will focus on the mTORC1 pathway, though. And the way we think about this is a pathway such as this has to sense two types of classes of molecules to make the decision of whether to grow or not grow. And one, of course, are nutrient levels. And I say, of course, now, although in the past, actually, it was not appreciated that higher organisms, multicellular organisms, needed in a cell-autonomous way to sense nutrients. We really thought that most of this happened through hormonal cues, such as insulin being the best example. But I think there's lots of evidence that this is the case now. So a cell in a multicellular organelle has to sense nutrients, know whether it can grow, and then also hormones such as insulin, plus a number of other ones, and make a decision as to both of these are present and therefore to drive growth. And we've been interested at the signaling level. How does this happen? How do you do signal integration? Now, we know a reasonable amount about this. A lot of it happens through a tumor suppressor called TSC, and this is an inhibitor of a small GTPase called Rheb, which Rheb is a direct activator of mTORC1. And so all of these signals through pathways which have components that are mutated in cancer, as you can tell, signals to Rheb. Okay, so this has been known for some time. Now, I would say that the molecular basis of this integration is not well understood. We've been interested in amino acids, as many of you know, particularly because this, I think, is a very primeval signal. And this is a signal that you absolutely need to have if you're going to turn on this pathway. And it turns out that amino acids probably do not go through this pathway, but go through an alternative pathway, which also ends in a different small GTPase. In fact, a heterodimer of small GTPases called the RAG GTPases. Okay? So it's a bit of a weird system. This one talks to this GTPase. This talks to different GTPases. Now, these proteins are quite interesting. They're related to RAS, like many of the small G proteins are, but very, very distantly. They have a C-terminal extension on them that we don't know the function of, which is not found in the proteins like RAS. And we have 4 of them, A, B, C, and D. And the most interesting aspect of these proteins is they come in 2 flavors, where you have A and B, which are very, very similar to each other, and as far as we can tell, redundant, and C and D, which are also very similar and redundant. And to make an active complex, you need an A or a B plus a C or D. And you'll see that I use these pretty much interchangeably. So what happens then when you perturb amino acid levels? And so we think what happens is when there's no amino acids, the A or B is bound to GDP and the C or D to GTP. You add amino acids, this flips in a mechanism that we don't know. And I don't mean literally that it swaps one to the other, although that is potentially a mechanism. And it binds now to mTORC1. And you can see a little bit of the data. This is quite robust. So minus plus amino acids, there's now an interaction. And if you make a mutant, in this case of B, that's locked with GTP, so it doesn't have hydrolysis capacity, you have a constitutive interaction. Now, it turns out if you express this mutant in cells, you trick those cells into thinking amino acids are present, even if they're not. So they become resistant to amino acid deprivation. And this is an experiment that's done by overexpressing this protein, which has potentially lots of caveats. And so we've done a much better experiment, which is to knock in this mutation into a mouse. And we didn't knock it into the B locus, because it turns out B in mice is not well expressed, but rather into the A, the RAG-A locus, which is widely expressed. And if you make meths from these cells, here are the wild-type ones, minus plus amino acids. This is one of our favorite outputs for mTOR. Actually, there should be a little P in front of that for phosphate, for phosphorylated residue. And you can see, though, the mutants have no signaling whatsoever, even though they express less of this protein. And this is not a mouse engineering issue, but rather it's a protein stability issue. This activated protein seems to be less stable. And I'll get back to these mice afterwards. These were generated by a postdoc, Alejo. So these proteins turn out to be necessary and sufficient to mediate this amino acid signal. And a lot of this has been published, so I'm not going to discuss it. But the real question became, how do these work? And I think this is where this aspect of signal transduction, for me, became much more interesting. It became more weird, and so therefore more interesting. So one of the clues to this came from images such as this, in fact, or the movies such as this, which what you're looking at is the localization of mTORC1 in cells that are deprived of amino acids. That's the starting state. And then we add amino acids. And what happens is that the mTOR is this white stuff. You see, you can start to see these little speckles that form. And they form very quickly, in very few minutes. This is very reversible. You can add back amino acids, remove them, and make these things or have them disappear. And so it turns out that what these structures are lysosomes. So what's happening is mTORC1 is going to the surface of the lysosome when there is amino acids and leaving the surface when there are no amino acids. And this is completely mediated by these RAG proteins. And in fact, those mutant cells that I told you about, if you look at the localization of mTORC1, it's always on the lysosomal surface. It does not care about amino acids anymore. And I want to stress, this is on the surface of the lysosome, obviously not inside of it, unlikely to survive inside. So this led to a model then, where we proposed that mTORC1 was in some unknown compartment when there's no amino acids. Rheb, we had shown, was on the lysosomal surface, and the RAGs in data that I won't show is also on the lysosomal surface. So we proposed that when you add amino acids, the nucleotide state of this complex changes in a way that's permissive to mTORC1 now binding to the surface of the lysosome, and now it can interact with Rheb and become activated, because Rheb is an obligate activator of this pathway. So this is a coincidence detector, right? You need these signals to GTP load this protein, and you need amino acids to put these guys into the appropriate nucleotide state to bind mTORC1. And so in this way, the cell can ensure that there's amino acids plus all of these other signals for it actually to make the decision to grow. And we've done a lot to try to prove this model, and I think to some extent we have lots of evidence that this is true, but I won't share any of that with you today because it's been published. But the main tool that we've used is an mTORC1 that we can force onto the lysosomal surface, even in the absence of amino acids. We've added a sequence that targets it to that surface, and then you get exactly what you expect. You don't have any inhibition when you remove amino acids, and the RAG proteins become completely superfluous, but Reb is still absolutely necessary. So there's a number of questions that come from this that we've been pursuing. One is this pathway deregulating cancer, and I'll show you a little bit of evidence to suggest that it is, although we don't yet understand how. And then the question that we're most focused on, what and where is the amino acid sensor? So it turns out the RAGs are not the amino acid sensor. We're fairly convinced of that. And I'll tell you up front that we don't know what the sensor is, although I think we're making progress in that direction. So the where, though, we're quite convinced we know where it is. And that is because over the years we've identified a separate complex that we've called regulator. This is a pentameric complex of quite small proteins, actually, that are shown here. This complex is tethered on the surface of the lysosome. And it turns out that as soon as we found some of these first components, particularly this P14, we were certain that we were on the right track because there's a human mutation in P14 that's a partial loss of function mutation, and it leads to the phenotype that's shown here. These are children that have both copies of this partial loss of function allele of P14. And these kids look relatively normal to you until I tell you that they're very small, so they're 1 percentile of their normal size. They have immunosuppression. They also look, I feel bad saying this, they also look really young in this picture. This kid, for example, is 18 and looks much younger. And these are all phenotypes that you get if you treat a mouse with rapamycin, particularly the immunosuppression and the growth defect. They also have a problem with pigmentation, and we don't really have an answer for why that might be. So this complex turns out to tether the RAGs to the lysosomal surface. It's required for them to be there. And more recently, I won't show you the data for this, but we've shown that this is also the GEF for RAG A and B. So it's an essential activator of loading A and B with GTP. So it has dual function. In fact, it's very similar to a complex you may have heard about called TRAP, which also has these tethering as well as GEF functions, although it has no homology to it. OK, so the model we have then is that all these components are on the surface of the lysosome. None of them are transmembrane proteins, to excuse how this is shown. And in retrospect, the idea that the lysosome would have an important role in amino acid sensing is perhaps not so far-fetched. This is an example, to me, of a discovery that when you look back upon it, you really think you should have figured it out before, right? If you sort of simply had thought about it enough, this would have made sense. And the reason I say that is because lysosomes are, of course, where autophagosomes fuse, where you get the payoff step of autophagy, the production of amino acids. mTORC1 is a major regulator of autophagy, so it kind of makes sense to have amino acids there where mTOR could sense them. In multiple organisms, for example, in the liver of rats, in the vacuoles of yeast, which are their versions of lysosomes, amino acids are known to be stored. Not all of them, but some of them are there. And so the idea that this is somehow sensing the amino acid state, whatever that might mean, of lysosomes was sort of our predominant idea in terms of what might be the connection of lysosomes to this pathway. Now, one question that you can ask that's simple, but turns out to be very, very hard to address, is where exactly are amino acids sensed? And there are two possibilities, right? One is on the outside of the lysosome, and the other one is in the inside, sort of an inside-out sensing mechanism. Now, this is hard to address in an intact cell, and so one of the things that Roberto, another postdoc in the lab, did was to develop an in vitro assay where he could take purified lysosomes, purified mTORC1, put them in a tube, add amino acids, and really, to our surprise, he got mTORC1 to translocate or bind to the lysosomal surface. And one of the things that this tells us is that all the sensing machinery is in purified lysosomes. Okay. So using this system, he asked a number of questions. So one of them is, do you actually need membranes? It seems a bit of a trivial question, but it turns out detergents don't break up these complexes, but you do need membranes to get signaling. Do you need the amino acids to go inside? And you do. And there's a number of ways that you can do this, but they have to be absolutely inside to get any sensing. And if you make little holes with, like for example, streptolysin O, you can't get them in there and you can't get any sensing. And then he asked whether the vacuolar ATPase, which all of you will know is the enzyme that pumps protons in to the lumen of the lysosome and acidifies it. And then that proton gradient is used by the lysosome to do many, many other things. For example, to bring amino acids in to the lysosome, as well as to, in some cases, take them out. So this turns out to be necessary. But the odd part is that if you got amino acids in, which normally the VATPase is necessary for, but if you then destroy the proton gradient with an ionophore, you still needed the VATPase. So the VATPase has dual function here as far as we can tell. One is to make the proton gradient to get things inside the lysosome. But then it has another function that we don't know what it is, but it's absolutely not the proton gradient. Maybe that's not so surprising because the VATPase is a very large complex that has at least, depending on how you count it, 12 to 16 components. So I want to prove to you that luminal amino acids matter inside an intact cell. The way we've done this is we have a way of very rapidly purifying lysosomes. They're tagged in this cell. We give them labeled amino acids, and this is a bead with little lysosomes attached to it, and we count the entry of amino acids into cells. And we do this in the presence of a protein synthesis inhibitor so we don't get tricked by proteins being made and degraded in the lysosome. So you can see if you add amino acids on the outside, within 10 minutes you find them in lysosomes. If you use detergents or streptolysin O, they're not inside there anymore. But most importantly, if you overexpress a protein called PAT1, you also block this accumulation. So we in the lab call this sort of emptying the lysosome of amino acids. So what is PAT1? So PAT1 is a transporter identified by others that exports amino acids out. It's completely lysosomal in its localization, as shown by this co-marker expression. And we don't actually think that this is part of this normal pathway. But for us, it's a very useful tool because I showed you just now that we can block the accumulation of amino acids in the lysosome. And so if luminal amino acids work, or how this pathway senses amino acid, this molecule should be highly inhibitory to the mTOR1 pathway. So this is the experiment, minus plus amino acids in a control situation. We activate this pathway. If we overexpress PAT1, we completely annihilate this system. Now, when you inhibit, you always have to worry, are you doing some toxic thing? But we have this trick that we can put in these activated alleles of the RAGs, and you can see not only do they rescue amino acid deprivation, minus plus is the same now, but they also completely rescue PAT1. So overexpression. So there are multiple potential interpretations of this experiment, but the simplest one in our mind is that you need to have amino acids inside to get this sensing to work. I should say one of the things that Roberto is doing now is doing metabolite profiling of isolated lysosomes to try to really understand what's inside of these things. And as you might imagine, it's not a trivial type of experiment. So the model we have then is there's no amino acids. mTOR1 is somewhere we don't know. You add amino acids, they get in. They probably can get in many ways, either by free amino acids or even potentially as proteins that get degraded through alternative sort of endocytic pathways. Somehow they talk to the VATPase, and I didn't show you the evidence for this, but the regulator is bound to the VATPase incredibly well, and actually we were surprised that people have missed this, and probably the reason is because the regulator are very small proteins. They probably were simply missed on gels when people were looking at what was on the VATPase. There's then a conformational change that activates the regulator GEF. This is then, of course, acts on RAG-A. And in a part that's much more speculative, we actually think this pops off the surface. Partly this is based on some of my colleagues saying, hey, David, mTORC1 is too big to diffuse around in time. It grabs mTORC1 and brings it down to the surface. Now you have this weird thing. This is over a megadalton, and this is 3 small GTPases are now bound to this thing, and it turns on, and you get growth. Now, I haven't said anything about the sensor because we don't know what the sensor is, but if I had to bet, I would certainly think it's the VATPase. And in fact, there are conformational changes that happen in the VATPase, complicated conformational changes that happen when you add or subtract amino acids. The problem is we've been unable to reconstitute this thing in vitro to really test that. And in fact, I don't think anyone has ever managed to reconstitute it. And so there's a big technical burden to proving this as a sensor. OK, so everyone says, hey, David, that's so complicated. And it is incredibly complicated. I don't know why it's so complicated. But the complexity might account for why this is a heterodimer, because this is something I haven't addressed, and nothing I've told you so far suggests that it has to be a heterodimer. So one of the things that we're thinking quite a bit about now is that compared to RAS, where there's really 2 states that can exist in, if you have this heterodimer, you actually have 4 states. And so we can define what 2 of these states do, but there's 2 sort of mystery states in here that we don't know what they do, and they could be involved in, for example, this shuttling of the RAGs from the surface back. And we actually do know, again, I'm not showing you this, we actually do know that this guy matters. What RAG-C is doing, what's stated in, is actually important for the activity of this pathway. So this is conceptually how we're thinking about this. Now this cancer connection. So every pathway that talks to mTORC1 has a mutation in cancer, every single one that we know about. And so the idea that the amino acid sensing pathway would not something that didn't make much sense to us. And so one of the approaches we've taken, there's a number, is to simply ask, can we find cancer cell lines that no longer can sense amino acids, just like that RAG mutant that I showed you. So here is what the vast majority of cancer cell lines look like. Minus plus amino acids, you turn off. When there's no amino acids, you add amino acids, you turn it on. Here is exemplars of 2 cell lines that don't do this. So minus plus amino acids are completely the same. And so we're using cells like this as well as many others and many different approaches to try to figure out what is wrong with these cells. And so it's almost certain that these cells are going to turn out to have mutations in the negative regulators of the RAGs. And we have evidence that that's the case, although there's probably multiple negative regulators. And so it's not going to be that all cells are going to fit in the same category. And so we're quite excited about figuring out what's going on here. Okay, I want to finish up just by telling you about this mouse that I showed you some of the cells for that mouse. This is a mouse which we knocked in this activated allele into the RAG-A locus. And we really had lots of fantasies about what we were going to do with this animal, because this animal will be resistant to amino acid deprivation. We thought it was going to have aging phenotypes. We're going to cross it to cancer models. We're going to do all this fun stuff with this animal. But we never got a mouse. So we got pieces of mice once in a while, but we never actually got a live mouse. And so we assumed that it was dying at some point during development. And so Alejo started doing time matings, going back and trying to look for when the embryos disappeared. And the bottom line is they never disappear. They're always in Mendelian ratios, no problem, even right before birth. And so he then delivered them by cesarean, and they were perfectly alive. And if you look at them, they're indistinguishable. Here are some mutants, here are some hets, some wild types. There's no difference in them. The only difference he could find is they weigh a little bit less. This is shown here. Histological examinations showed no differences. And so at the end, he had to conclude that these mice were dying after they were being born, and the mothers were eating them. So they had a perinatal death. And in fact, when he went and delivered them by cesarean, put them in an incubator, and asked what happens to them, that's exactly what happened to them. So here are control mice, hets and wild types. This is very stereotypical. This has been done in many different mouse strains. They die, all of them, by 24 hours in this type of situation. So they have water, humidity, and food. Sorry, not food, and heat, but no food. And no mother. The mutants die in exactly about half that period of time. Now you could say this is some developmental problem that only manifests itself once they're born, and so you really don't know what's going on here, even though we found no defect in these guys. But you can actually rescue this completely by giving them rapamycin. So if you just inject them with rapamycin, you can rescue them so these lines are practically overlapping, which really tells us that the mTOR1 inhibition is like hyperactivation, which I'll show you afterward, is a problem in this system. So many of you will know that this phenotype of not surviving perinatal state, being a little bit smaller, is highly reminiscent of mice that have defects in the autophagy pathway. So if you delete ATG7 or ATG5, which are key autophagy genes, you get exactly this same phenotype. If you go into these mice and ask, can they trigger autophagy, the answer is no. I'm not going to show you that. So you can do that by EM, by LC3 cleavage, whatever you want to do, you can show these mice cannot trigger autophagy, which has been argued that in this perinatal state, you have to trigger autophagy to get food, get nutrients to survive before you suckle. And this is particularly true in a mouse, because a mouse has very little fat compared to a human. So a mouse is a little bit more like a premature human. So now the output of autophagy that matters here, and what I will show you, are amino acid levels. So if you look at amino acid levels, either branch chain or essential, right at birth, there's no difference. If you look 10 hours later, you can see there's a severe defect in the amino acid levels of the mutant mice, the GTP mice. Okay. And so that's what you would expect if you can't break down protein to liberate amino acids. Now this is quite late in their life, actually. These mice are going to die relatively soon. And so we asked, well, what is the mTOR1 pathway doing earlier, for example, at this time point? Can we try to figure this out? So this was a little bit surprising to us. So if you look at when they're just born by cesarean, you can see there's a little bit of noise and mTOR1 signaling, as you might expect. But by 1 hour, the mutant is still active, while the other ones are really very much off. This is true in the liver. This is also true in the heart. And it's also true in any system that we've looked at. So this means that something drops within 1 hour of life that mTOR1 normally senses through the RAG pathway but can't be sensed in these mutants. Okay. So the obvious candidate was amino acids. So I showed you amino acids were down by 10 hours. But if you look at by 1 hour or around that time point, they're actually not down that much. They're down a little bit, but not down particularly. And so this was a little bit confounding to us. What was being sensed here that the RAG mutants could not sense? So we looked at glucose, and this is what glucose does. So at birth, this is the amount of glucose in the mouse. This is actually where I know this is the glycemia, mg per deciliter. You can see it plummets by 1 hour. This is a high overestimation because this is the limit of detection of the glucometer. In fact, if you measure it other ways, it seems to almost go to 0. It then bounces back quite nicely and eventually comes back down. And eventually, when this crosses this line again, this is when this mouse dies. Now, the reason it comes back up is that it uses those amino acids that I showed you before in the blood to make glucose in the liver, to do gluconeogenesis, and it also breaks down the glycogen that it has to release glucose. Eventually, you can't break down amino acids anymore, and you run out of glycogen, so this is an unsustainable situation for this mouse. Okay. Now, what happens to the mutant? The mutant is identical in this early period, but you can see it never recovers. Right? It can never raise its glucose levels again, partially because likely it doesn't have the amino acids to make glucose. We can rescue this. If we give these mice just glucose, we can rescue their lethality. And if we just give them amino acids that can be used to make glucose, but not to make all proteins because we're just giving a subset of amino acids, we can also rescue them. So clearly this defect in glucose levels is not just a correlation of the lethality of these mice, but actually is causal in their lethality. Now, what I just told you, though, should be a little weird to you, because I just told you all this sort of stuff about this is an amino acid sensing pathway, and now I'm telling you, well, glucose levels by 1 hour drop practically to 0, and yet this pathway does not care, right? And so it still should be sensing glucose. If what I showed you before about all these two inputs to this pathway, this does not make sense. And so we had to reassess this model. We always assumed glucose was sensed as an energy store through ATP levels, and that is certainly true in many situations. Through AMP kinase is one of the key pathways. But we're now thinking that glucose is also sensed, perhaps not as an energy store, but rather as a nutrient through this RAG pathway. And in fact, I should remove this arrow because we know that this is true. If you take mutant cells and you remove glucose, they're incapable of sensing it. Moreover, if you make media that mimics the plasma concentration of nutrients that you find in a starved animal, these cells cannot sense it. So that combination of nutrients is unsensible if you have an activated allele of this protein. So one of the things that this is telling us is that at least in this perinatal state, we think of these organisms as quite simple. They're a little bit more like yeast. They're sensing directly nutrients, amino acids for sure, likely glucose, as I told you. And it's already known that in the perinatal state, things like insulin don't matter. So insulin, in fact, is not even really produced until you start to suckle. Right? And so these are sort of primitive nutrient sensing situation in perinatally. So what we think happens then is right after birth, there's glucose, this pathway is on, autophagy is off. But by 1 hour, glucose drops, this pathway is inhibited, autophagy is on, amino acids are released, probably from the muscle, but probably from a number of different tissues. We actually have found very little difference in autophagy induction in many different tissues. The glucose takes those, makes glucose comes back, this pathway then you can turn this off. Now, in the mutant, though, you never sense the absence of glucose. You never trigger autophagy. And so you can never go into this kind of cycle. OK, so the conclusions really is that the big ones are that this is an inside-out amino acid sensing pathway. Obviously, we want to find out what the sensor would be. And I'm not saying that only from the inside out, but that we know there's got to be a sensor there. And then lastly, we were quite surprised that by making a simple point mutation in these RAG proteins, we have this severe defect in viability in this early part of life, and we have a severe defect in autophagy. And the reason I say we were surprised is if you look at any of the reviews on autophagy, you see a huge number of pathways that are supposed to trigger autophagy. Some are mTOR dependent, some are mTOR independent. And yet here what we've done is we've changed a single component just upstream of mTOR, and we seem to have a severe defect on autophagy. So Yasmin was a student who originally found the RAGs as part of the mTOR pathway. Liron found the regulator. Lynn and Liron are now working on sort of this cancer connection that I alluded to. Roberto really pioneered all the work on lysosomal signaling, is doing a lot of metabolite profiling on lysosomes to try to really understand them as a sensing organelle. And Alejo made the mouse that I told you about at the end. And so if there's time, I'm happy to take any questions. Thank you.